Hey everyone, and welcome back to Science Unpacked. Today, we're diving into a fascinating paper about enzymes, specifically one called galactose oxidase.
Ooh, enzymes. Always doing amazing things in our bodies and in nature. What makes galactose oxidase so special, host, eh?
Well, it's a copper containing enzyme that's crucial for converting alcohols into aldehydes. Think of it as a tiny, efficient chemical factory.
So, like transforming one type of organic molecule into another? That's pretty fundamental chemistry, right?
Exactly. And its active site, where all the action happens, involves a copper ion and a very reactive phenoxyl radical. These radicals are tricky to study.
A radical? That sounds a bit unstable, doesn't it? Like something that wants to react with everything?
Precisely. And synthesizing stable models of these radical containing active sites has been a real challenge for chemists. They usually need external oxidants.
So, this paper found a new way to make these complex enzyme models without all the extra steps?
Yes, of facile one-pot synthesis. They created two copper complexes. Complex 1 is a non-radical, but complex 2 is the star, a phenoxyl radical complex.
A one-pot synthesis for a radical complex? That's a big deal. What makes complex 2 stable enough to be a radical?
It's all in the ligand design. Host B, complex 2's ligand, has these bulky tertiary butyl groups. They act like little molecular shields, stabilizing the radical.
Ah, so the structure itself helps protect that reactive radical. Very clever. How did they confirm these intricate structures?
Through X-ray crystallography, which gives us a 3D atomic blueprint. They could literally see the copper and the ligands arranged.
That's incredible precision. And to know one was a radical and the other wasn't, did they use special tests?
Absolutely. They used UV visible spectroscopy and EPR, which is like a fingerprint for radicals. Complex 2 showed a clear radical signature.
So, they've built and characterized this amazing radical complex. But does it actually do what the enzyme does?
That's the next exciting part. They tested its ability to oxidize benzyl alcohol. And guess what? Complex 2 was significantly more effective than complex 1.
So, it's not just a pretty structure, it's a functional mimic of the enzyme. How does it perform this oxidation?
They propose a hydrogen atom transfer or HAT mechanism. The phenoxyl radical basically snips off a hydrogen atom from the alcohol.
Hydrogen atom transfer, so it's like a tiny molecular hand grabbing a hydrogen? That's a very specific action.
Exactly. To prove it, they showed complex 2 could convert 9,10 dihydroanthracene to anthracene, a classic HAT reaction.
That's solid evidence for the mechanism. Did they discover any other surprising abilities of this radical complex?
They did. Complex 2 also demonstrated self-activated DNA cleavage. It could break down plasmid DNA without any external activators.
DNA cleavage? That sounds like something out of a sci-fi movie. Is that a good thing in this context?
Potentially. Yes. Controlled DNA cleavage has huge implications for gene therapy or targeted drug delivery. It's a powerful tool.
So, this single complex could be used to precisely cut DNA strands? That's truly groundbreaking for bio applications.
It certainly is. And they used DFT calculations to understand the electronic structure, confirming how the radical and copper work together.
This paper really brings together elegant synthesis, detailed characterization, and exciting functional applications. It's a comprehensive study.
Absolutely. It's a significant step forward in mimicking complex biological systems in the lab, and with simpler synthesis too.
And that simplicity means it's more accessible for other researchers, potentially speeding up future discoveries. What a brilliant piece of work.